Here's what you'll need: spinach leaves, baking soda also known as sodium bicarbonate, water, liquid dish soap, 200 ml beaker, spatula, hole punch, a 10 ml syringe without the needle, color cellophane sheets, light bulb, timer, paper and pen to record the results. First thing, I'm going to make 0.2% solution. This is not very much, it's only about 1/16 of teaspoon of baking soda in 150 ml of water. Then add 1 drop of liquid dish soap to the solution, and I'm going to mix that. For this experiment, I will use spinach leaves with a hole punch. I punch tiny round pieces, and I need 10 leaf discs for each trial. If I drop a leaf disc into the solution, it will float. This is because some air trapped in the layers of the leaves. I need to force the air out of the leaf disc using a syringe. If I hold the tip of the syringe close and pull out slightly on the plunger, I create a partial vacuum inside the syringe. With a partial vacuum inside the syringe, the air pressure inside the barrel is much less than the pressure outside the barrel. When I release the plunger, the outside pressure forces the plunger back to its former position. Now, I will remove the plunger from a syringe and dump 10 leaf discs into the barrel. Next, I insert the plunger and force as much air as possible out of the syringe by depressing the plunger almost to the bottom of the barrel. I need to pull a small volume of sodium bicarbonate and soap solution into the syringe. By closing the tip of the syringe and pulling on the plunger, I create a partial vacuum between the surface of the solution and the end of the plunger. Air trapped in the leaf disc will flow into the partial vacuum. After pulling on the plunger, I hold it in that position for 10 seconds. Then, while still holding the tip of the syringe, close and slowly release the plunger to its former position. This was done until all the leaf discs sunk in the solution and were ready for testing. Next, the leaf discs and solution were then put back in the beaker. The beaker was placed under the light with colored cellophane sheets, and the number of leaf discs floating were counted and recorded at 1 minute interval for 25 minutes. Once all the discs were floating or when 25 minutes were over, the next test was completed. The following data table displays the number of leaf discs that had risen to the surface of the mixture by each minute of the 15-minute trials. The graph shows the rate of the leaf discs rising for all the colors. As you can see, the blue leg had the most discs rise to the top by the end of the 15-minute trial, while green leg had the least.